Okay, so hopefully you guys have open right now in front of you the grid-in practice questions that can be accessible through the updates of your Schoology page. And so the very first color talks about sex-linked traits in Drosophila melanogaster, which is fruit flies. It tells you that red eyes, so capital R, is dominant over to white eyes. Shh. And so this is sex-linked, so it's going to be always on, well, for the most part, the X chromosome. In this class, all of our sex-linked crosses, the trait's been on the X chromosome. Every Punnett square you want to set up XX crossed by XY, right? That's your basic fundamental. If it mentions sex-linked, this is what you want to do. And then let's talk about the trait. Well, the female was heterozygous, so now we do heterozygous, big R, little r. And the male was red, so you could only put the red allele on the X. Nothing goes on the Y. And so you got big R, that's a female, that's red. This is big R, little r, that's a female, that's red. That's big RY, that's a male who's red. And that's little RXY, that's a male who's white. So if you look at it, 75% of the offspring are going to be red-eyed. And this guy right here is white. So we have a 3-to-1 ratio of red and white to white. Okay? And so that's what we expect to see. So your expected frequencies are going to be derived from what you get on the Punnett square. And so they tell you that your observed of red and white, here's the data table they gave you. Red, they said 134 flies. It doesn't matter if they were guys or girls, were red. And 66 flies were white. And so that's a total of 200 flies. That's the magic number we need, the total number of flies. The thing is when you're doing a chi-square analysis, you need to compare observed minus expected, right? And so there's usually like a T-chart you want to create. And so your observed of the traits red and white eyes were 134 and 66. So how do I calculate expected? Well, that's based on my probability in the Punnett square of what I should have seen. And so it's a 3-to-1 ratio red to white. It is 75% red and 25% white. So make sure that, how do I find this number? I need to use as the total number of flies that were observed. You take the expected ratios and multiply it by the total number observed. You're always going to get it right if you don't put actual numbers of organisms in the expected. It's never a percentage. It's never a frequency. It's the actual number. And so you should have seen 150 red eyes because that's 75% of 200. And you should have seen 50 white eyes because that's 25% of 200. And so there you have it. Now you can do a chi-square analysis. And chi-square is pretty simple, right? It's the sum of the observed minus the expected squared over the expected. And so if you were to actually go and figure out if you had to accept or reject the null hypothesis, you would need to know the degrees of freedom, which in this case is what? 1, because we're only comparing two data outcomes. And so 2 minus 1 is going to be 1. And then you would go into the table, and then you'd find what the critical value was, which is 3.84 based on 0.05 or 95% confidence. But they're not even asking for that. They just want to know the chi-square results. What is the chi-square value? And so you just take 134 minus 150, square it, divided by 150, and then you add that with 50 minus, 66, the observed, 66 minus 50 squared over 50, right? And that's going to equal, once you do all the math, let's see, that's going to equal about 6.83. Okay? Because this is going to be right here, this is going to be around 5.12, and this is going to be around 1.70666. Okay? Here's the key, guys. If you can, what's best? Always try to round at the very, very, very end. Leave your numbers as long as possible, right? Like carried out to the 17th decimal point, if you can on your 4-function calculator, and then just do all the rounding at the end. So if your four-function calculator is giving you a number that has 5 or 6 decimal points, leave it like that until we add and subtract and divide till the very end, and then do the rounding. Do the rounding at the end, okay? Because it's going to be best that way. Okay, so the next one is surface area to volume ratio of a sphere. And so they give you, based on the shape, equations of surface area and equations for volume. And so they're telling you to find the surface area to volume ratio. So ultimately what you're doing is you're going to take whatever the surface area is and you're going to divide it by the volume. And that's going to give you the surface area to volume ratio, correct? Well, for a sphere, surface area, or area, is, if you look at the equation sheet, surface area is 4 pi r squared. And volume is 4/3 pi r cubed, right? Okay. Now, if you look at it, surface area divided by volume is the same as surface area multiplied by the inverse of the volume. The volume is 4 pi r cubed over 3, correct? So the inverse of this would be 3 over 4 pi r cubed. So the short way is to do this. The surface area multiplied by the inverse of the volume. 4 pi r squared multiplied by the inverse of the volume, which is 4 pi r cubed. Everybody see that? Look at what cancels. 4 is going to cancel out. Pi is going to cancel out. And 2 of the r's are going to cancel out, leaving behind 1 r. So really, it's 3 over r, right? The surface area to volume ratio. R is 5. So it's 3 divided by 5. Round to the nearest hundredth. What's 3 divided by 5? There you go. Round to the nearest hundredth, so it's going to be 0.60. Please make sure you include that last 0. Don't put 0.6 because that's the nearest 10. Next question is going to be a negative I CRT question, and they give you what R is, which is the pressure constant at 0.0831. So if we're calculating the solute potential, remember it's an open beaker, so the pressure is going to be 0, right? There's no pressure. It's an open beaker. The water potential is equal to the pressure potential plus the solute potential. But if you're dealing with an open beaker, there's no pressure potential, so the solute potential and water potential are equal. And the solute potential is equal to negative I CRT. Well, so the I really has to do with what type of solution it is. If it's a sugar solution, sugar can be considered one of two things. Sucrose, which is a disaccharide of glucose and fructose, and glucose is also we call sugar, right? Glucose, sorry, C-ose, which is just a monosaccharide of glucose. Either way, when you put either one of these into water, they're going to stay together as one thing, right? So the ionization constant of both sucrose and glucose should be what? 1. If it's talking about a salt solution, it's most likely talking about NaCl, and when you place that in water, it turns into sodium ions and chloride ions, and so the ionization constant of salt should be 2. Unless they give you a really big curveball and tell you that the salt solution is calcium chloride, in which case the ionization constant would be 3, because it turns into 3 ions. Now, I don't know exactly what that's going to be, but know this. The ionization constant of sugar is 1. So this is a pretty easy math equation when it boils down to it. It's negative 1 times C, which is the concentration of the solution, which in this case is 0.3 molar. It's in the units of molar. The R, which is always 0.0831, that's a constant. It's provided with you on the formula sheet, so it's 0.0. Please, somebody be handy with a calculator, so they're doing this for me. 831. And the temperature is in Kelvin, so that's going to be 273 plus the Celsius. So 273 plus 27 is going to be perfectly 300. So when you multiply all those guys together, you're going to end up getting 7.5? Not 7.5. Negative 7.5. Right? Because don't forget that negative I. If you put 7.5, you will get 100% wrong. Because this is like a multiple choice question. It's worth a point. So your answer, you make sure that you do not forget to bubble in that negative. and then put in 7, and then put in the point, and then put in the 5. And because it said nearest 10th, right there it said nearest 10th. It didn't say nearest 100th, it said nearest 10th. All right, so water potential is usually written in with this psi, which equals bars. And 1 bar is equal to 1 megapascal as well. And so don't let the units throw you off, right? Remember that the water potential is equal to the pressure potential plus the solute potential. So this is telling you that you're inside of a plant in the desert. And so the plant's internal pressure potential, right? So the what's causing water to go in and out based on just pressure, hydrostatic pressure, is negative 0. I'm sorry, the solute potential. The solute potential based on what is dissolved in the water is negative 0.0. Oh, sorry, negative 0.6. My bad. And the pressure potential is negative 1. So negative 1 plus negative number is the same as subtracting a number. So this is going to be negative 1.6. So the answer is negative 1.6. Everybody see that? Pretty easy. That was, and it's to the hundredth, so it's actually negative 1.6 what?
0.
Negative 1.60. All right, so make sure you do it to the hundredth. My bad. Please don't miss the question. If it says round to 1 decimal point, if the answer is 5, like it was on your practice test, please write it as 5.0. Yeah, because it was wrong. Because 1 decimal point is a tenth. So if you put the number 5, you would have gotten it wrong. Because it asked you not to the nearest whole number, it asked you to the nearest 1 decimal point, which is the same as saying to the nearest tenth. So please round exactly as it asks you to round, and please, if you can, save your rounding to the end. So this is a Hardy-Weinberg equation. Guys, all Hardy-Weinbergs, the mass, you typically want to find out what this is, which is the genotypic frequency of the little a's. So if typically they're going to tell you a problem that gives you the number of individuals that are of the recessive genotype. And so they told you that. There's some sort of beak condition where 24 of the birds had the recessive condition, and 63 of the birds had the non-recessive condition. So pretty much what they said is big A, and big A, little a equaled 63 birds, and the individual birds that were little a is 24, for a total of how many birds total? 87. Guys, we always want to know total number of organisms in any sort of biological math problem. The number of the population is always important. So you got to find Q squared. Now, is that 24? Nope, it's 20. It's an allele frequency. It's a genotypic frequency, so it must be in a percentage or a decimal point that represents a percentage. So it's not 24 divided by 63. It's 24 divided by the total, which is 87. So out of 87 birds, 24 of them had the recessive condition. That is going to give me 0. What? So that's going to give you 0.276. That represents Q squared. It's asking me to find the frequency of the dominant allele. I want to know what big A is, and remember, big A represents P, and little a represents Q. So in order for me to find P, I need to know what Q is, and 0.276 is not Q, it is Q squared. So I take the square root of Q squared, square root of this, and that will give me Q. Right? That's Q, which is what? What's the square root of 0.276? 0.525. So Q equals 0.525, right? So P is going to equal, remember, P plus Q equals 1, 0.475. So you're going to round up P, because it wants it in the hundredth, to what? 0.48. You put 0. Or you can shift it over to the right one. All right. I guarantee you there will be, let me get a little room here. There will be some sort of problem that has a data table that contains how much something is consumed or produced over the amount of time. Guarantee you. And then they're going to ask you to calculate a rate but they're going to give you a specific time frame that they want you to calculate the rate. So really what you need to do is you need to take the final rate minus the initial rate and divide it by the final time minus the initial time. It's really simple. So they're telling you in between 2 and 4 minutes, so this is the only data that matters, in between 2 and 4 minutes. You want to take the final amount of oxygen produced, 5.5, subtract it from the initial, 3.6, divided by, because this is a rate, remember, so it's going to be how much is produced or consumed over time. The final time, which is the 4th minute, minus the initial time, which is the 2nd minute, and that is going to give you 1.9 divided by 2, which is going to give you 0. And they want it in hundredths, 95. That is the answer. You don't need to know units because it's a gridded. So why would they have a question here for you guys to practice? What is the probability of doing 3 coins simultaneously? What does that have to do with biology? Well, when it comes to genetics and you're given genotypes of parents that have like big A, big B, little c, big D, little d, big E, and it's crossed with another parent that has, 5 different genes in their genotype, you're not going to do a Punnett square with 12,000 squares. You're going to use the laws of probability in order to determine what is the possible offspring's genotype or phenotype based on the parental genotypes. And so, what are the chances of throwing 2 coins getting heads 3 times? Well, what are the probability of getting heads 1 time? It's 1/2. Doing it again, you multiply the probability again. Doing it again, you multiply the probability again. Expressed in fractional form, you would do 1/8. You can do like this, 1 divided by 8. That's one way to do it. Or you can do the 8 over here, and then the division sign right there, and then the 1 sign right there. That would work too. Okay, per capita growth rate is how many individuals are added to a population over a given time. Not how many people are born. How many people join the new population after a period of time. So in order to know how many people are added, or subtracted for that matter, you have to take in the birth rate, how many people were born over that time period, and you have to subtract it for how many people died. Now, if more people died than more people were born, then you're going to have a per capita growth rate that is in the negative. If more people were born than that died, then you're going to have a per capita growth rate that's in the positive. And so the way that you calculate birth rate and death rate is you take how many people were born over the total population, and then how many people died over the total population. And that's going to give you a birth rate or death rate. They tell you that our starting population is 2,000. And 1,000 mice are born. So can I use that information to calculate a birth rate? Yes, I can. Birth rate is 1,000 people were born that month. Out of 2,000, the birth rate in terms of per month, each month is 0.5. That's the birth rate per month, 0.5. It's added. You add 50% of the population every month. Death rate, well, look, 200 mice died. So we got 200 mice died out of 2,000. So the death rate equals 0.1. It's 10%. So they want you to find the growth rate. Well, it's the birth rate minus the death rate. So it's 0.5 minus 0.1, which equals 0.4. Nearest tenth, yep, 0.4. Is that hard? Absolutely not. You just got to know what it's asking and how to calculate that. Yep. I would do it just in case. Might as well. Yeah. Right here, where it says AP Exam Review. Free response. Oh, by the way, well, I'll show you guys that in a minute. The free response questions. Here's a link to 2000 and some free response questions and guidelines from 2013, 14, and 15. And we're going to go over some of those today after I'm done. But right here, how to answer grid-in. Okay, so if you want to know how to answer grid-in, there's one right there. Just some questions on how to do it right there. I threw that in there for you. Okay, so I would just always put in the 0, might as well. Well, if you're putting the too many zeros after the decimal point and you're taking it to the hundredths and the thousandths when it's asking for it in the tenths, then you're going to get it wrong. So please put it to the zero. If you put in 2 zeros beforehand, why are you doing that? No. Okay. So this one tells you that the primary productivity, the net primary productivity, right, is 8,000 kilocalories per meter squared. That's the NPP. It wants to, if the respiration of the aquatic producers, that's the R, is 12,000, then what is the gross? It's telling you that it wants to know what was your paycheck, right, if you took out $12,000 in taxes and you were left with $8,000. That's really what it's asking you. It wants to know what is the gross annual primary productivity per year. Round to the nearest whole number. Well, solve for X. Add 12,000, right? Add 12,000. So the gross productivity should be 20,000 kilocalories per meter squared, but you don't need to put that. That's your primary productivity answer. Okay. Okay. So the next question, people were getting a little nervous about, because they didn't understand what is the temperature quotient, because we really didn't go over it. There was an old lab where you took a fish, and you put it in like lukewarm water, like regular water. So let's just say, I don't know, 23 degrees Celsius, right? And then you measured its heart rate, right? Its heart rate per minute, right? Heart rate per minute. Let's say, and you could do that. We didn't use a fish. You can do that by how much the gills are flapping open and up. But there's also these other little organisms called Daphnia, and you can actually see their heart beating, so you can count. And then what happens is you would put it in 10 degrees colder Celsius. So, sorry, this would be 13. Or you would just change the temperature. 13 degrees Celsius, and then figure out what the heart rate was there. Oops, the heart rate. Or if you put it in warmer water. Right? Same thing here. If you put it in warmer water and figure out what the heart rate is here, and see how much did this metabolic process increase or decrease by when you increase the temperature by 10 degrees Celsius or decrease the temperature by 10 degrees Celsius. Did dropping the temperature 10 degrees Celsius slow it down or speed up the process? And in most cases, it slows it down. And did increase in temperature, right, by 10 degrees or however much, increase, and by how much? And that's what the temperature quotient is. The temperature quotient tells you how much the metabolic process would increase or decrease. Let me give you a little bit of sample data, and then I'm going to give you this data. So that's what Q10 is. Q10 equals the second rate divided by the first rate. 10 to the 10th power, so let me get rid of this, to the 10th power divided by time temperature 2 minus temperature 1. Let me give you some sample data. Let's say that you had, this is heart rate over here, and this is temperature over here. Let's say at 20 degrees Celsius, the heart rate was 8 and something, right? And at 30 degrees Celsius, it went to 16. And so if you plug that in, your K2, right, your K2 is 16. That's K2. Your K1 is 8, right? And then it's 10 divided by 30 minus 20, which is the same as 16 divided by 8 over 10 over the 10, or 10 to the first, which is 1, right? And so the Q10 in this case is what? 2. What this means is that when you increase 10 degrees temperature, the metabolic process doubles. That's what the temperature quotient. Is that what we see here? Right? Using this math, the Q10, or the temperature coefficient, is what would happen to the rate of that metabolic process when you change the temperature 10 degrees in either way. A positive 2 means it doubles when you increase temperature, right? And a negative would be like it would decrease. So, let's look at your sample size. Your K2 is 22. Okay, remember this is the over K1, 10 divided by temperature 2 minus temperature 1. So K2 is 22 over 16, right? And this is 10 over the second temperature, 21, minus the first temperature, 16. So that's the same as saying 22 divided by 16, 10 over 5, which is the same as what? Squaring it, right? So it's 22 over 16 squared. It wants you to round to the nearest whole number. Okay? So somebody want to do that for me real quick? If you do this, this is going to be 1.890. It rounds to the nearest whole number. This is 2. So the temperature coefficient is, round to the nearest number, it's 2. It's not asking to the nearest decimal point. So this guy is on pace to pretty much double the rate of respiration every time you increase 10 degrees Celsius. It's kind of on rate for that. All right, standard deviation of 1, 2, 3, 4, 5, 6 data points. Okay? So standard deviation equals, if you look at the formula, I'd like everybody to sit here, since everybody's falling asleep, and give me the standard deviation of this one, and then I'll double check to see if you guys are right. The standard deviation problem that is looking at is the square root of the sum of xi, which is each individual data point, minus x with the line over it, which is the mean, squared, minus the mean, over n, which is the number of data points, minus 1. First thing you guys got to do is calculate the mean of those 5. Okay? First you guys got to calculate the mean, which is 2.0 plus 2.2 plus 2.1 plus 2.0 plus 2.4, right? All divided by 1, 2, 3, 4, 5. Okay? So hopefully everybody should have gotten, what, 2.2? Okay. So now let's do the standard deviation, which is the square root of, let's do the first data point, 2.0 minus 2.2 squared, plus 2.2 minus 2.2 squared, plus 2.1 minus 2.2 squared, plus 2.4 minus 2.2 squared. I'm running out of space here. The last one is 2.5 minus 2.2 squared. All of this is divided by n minus 1, and since there's 5 data points, n minus 1 equals 4. Am I? Am I missing a second? I am. So 1, 2, 3, 4, 5. Oh, there's 6. There's 6 data points. My bad. This is 6. My bad. So I'm missing a data point. That should be squeezed in here. So this is actually 5, so my bad. It's still going to give you the mean of 2.2. That should be 5. That's actually 6. So I'm missing a 2.1. 1, 2, 3, 4. I'm missing a 2.5 here on up here. And then I'm missing a 2.0 here. Minus 2.2 squared. Right? So there's your math problem. Once you add up all these on top and divide by 5, please don't forget to square root. So you end up getting what? Does anybody have the final? What'd you get, Sam? 0.21. So 0.21 because it asks for it in the 100. There's your standard deviation. Ben has a 2 molar sucrose solution. He wants to make 3 liters of 1 molar sucrose. How much of the original solution should he dilute? Concentration of the first solution multiply the volume of the first solution should equal the concentration of the second solution. Multiply the volume of the second solution. Concentration of the first solution was 2 molar, right? Multiplied by the volume, which is what we're trying to find here. That's X. Concentration of the second is 1 molar multiplied by 3 liters. Right? And so you divide by 2 molars here. And this is 3 divided by 2 molars. Molars are cancel out, and then you're gonna end up getting X equals 3 liters divided by 2, x equals 1.5 liters, which makes sense since you want to take a solution that's twice as concentrated and make it half as concentrated. You're only going to need half of the solution to fill the rest with water, right? And that's going to give you your 3 liters. So the answer is 1.5 because it's asking for the nearest tenth. This is a lot simpler than, this is going back to chemistry, so we didn't really spend a lot of time, but it's your negative log concentration of hydrogen ions equals the pH. So if the pH is 2 and you got negative log, how do I find H plus? What do I got to do? 10 to the negative 2 is going to give me that. 10 to the negative 2 is 0.01. Right? And that's the nearest 100. That's the concentration of hydrogen ions, something with a pH of 2. Alright, so this is Artie Weinberg. Okay, you guys can start, I would probably start making some little signals here. We got sickle cell anemia. To not have sickle cell, that's P squared, right? Obviously, this is normal. That's your P, dominant. And then they're using an S instead of a little a as your Q. This is your 2PQ, the heterozygotes, and this is your Q squared. We know that if you have sickle cell anemia, or if you're heterozygote for sickle cell anemia, it is much harder for you to get malaria. They're telling out of 1,000 people, the key here, guys, is 1,000 people, 250 of them are homozygous for sickle cell. We always want to start with that Q squared. So Q squared, do I put 250 here? No, it's got to be a frequency. It's 250 out of 1,000. So Q squared, which is a genotypic frequency, is 0.25. But I want to know, okay, how many people in the population are, how many actual people out of 1,000 should show increased resistance to malaria? Round to the nearest total number. So how many people are, right, little s, and big A, little s. So in order for me to do that, I need to get P. I need to calculate for P in order to calculate 2PQ and then multiply that by 1,000. So in order for me to get P, I need to take the square root of Q squared. And so Q equals 0.5, right? And P equals 0.5, because you subtract from 1. And so the number of these individuals, heterozygotes, is going to be 2 times P times Q, which is 0.5. So if 25% of them, or 250 of them, are going to be this is 250, how many are going to be this? 50% are going to be this. That's the 2PQ, so that's 500. So 750 of the individuals, because first all I had to do was calculate genotypic frequency of 2PQ and genotypic frequency of Q squared, and then multiply that by the total individuals, and that got me 250 and 500, even though they already gave me the 250 number. Okay, so the answer would be 750. Pretty simple. All right, so this is a dihybrid cross. Remember, if I take big A, big B, little b, and I cross with big A, big B, little b, you're going to get a 9 to 3 to 3 to 3 ratio phenotypically. 9 of them are going to be dominant for both traits, right? Dominant, dominant, okay? That means they're going to be, whatever big A allele is and big B is. 3 of them are going to be dominant for one trait and recessive for the other. That means you're going to have whatever the allele for big A is and little a. And then 3 of them are going to be recessive for first one and dominant for the other. So that's like a little a and a big B. And only 1 out of 16 is going to be recessive for both traits when you do a dihybrid cross. That's little a, little b. Now, that's that 9 to 3 to 3 to 1. That's what you expect to see. So they're telling you that in flower colors, purple is dominant over yellow. Okay, so this is the dominant and this is the recessive. And then smooth of seeds is dominant over wrinkled. So we expect to see 9 out of 16 purple smooth. we expect, so this should be 9 out of 16. Purple wrinkled is dominant recessive. We should expect to see 3 out of 16. This is recessive dominant. We should also expect to see 3 out of 16 for these. And then yellow wrinkled is recessive for both. We should expect to see 1 out of 16. And so we should expect to see these frequencies, this 9 to 3 to 3 to 1. So it gave us actual observed. And so your observed was, of the purple smooth, 233. This is the dominant. 84 of the dominant and recessive. Right? Sorry, thank you. 223, thank you. 89 and 33. Okay, of the recessive dominant and then the recessive. What does that all add up to, guys? Because remember, the most important thing of you guys when doing all these biological problems is you need to find the total number of individuals in the observed. If you add all this up, what do you get? You get 429. That's the magical number. That's the magical number, the total number of flowers, right? Or seeds, in this case, kernels. So how do you calculate expected? Well, you take 9/16 and you multiply it by 429. And so what is 9/16 divided by 429? What is 3/16 divided by 49? What is 3/16 divided by 49? What is 1/16 divided by 49? Take a minute and give me those 3 expected numbers. Don't round now. The expected is 241.81. Oh, sorry, 3125. Yeah, I'm looking at a different one. Sorry, 3125. That's a 3. And then this one is 80.375. Sorry for my penmanship, guys. And this one should be the same, 80.4375. And this last one should be 26.8125. Okay? So when you set up the chi-square, you're going observed minus expected. So 223 minus 241.3125 squared over expected 241.3125. Okay? I would just round it at the end. And then plus the next one, right? Which is observed 84 minus 80.4375 squared divided by 80.4375 plus the next observed, which is 89 minus the expected, which is 80.4375 squared over 80.4375 plus the last one, which is 33 plus 26.8125 squared divided by 26.8125. It's just a lot of jumbled mess, and I apologize, but once you add all that, you're going to round to the nearest hundredth. Okay? And that'll give you your chi value, which should be, let me see if I have the answer somewhere. The answer to that I can't remember what it is off the top of my head. Okay, this is going to be delta G equals delta H minus the temperature delta S. So if they tell you, right, that in the unfolding of a protein, the delta H or enthalpy is 46,000 joules per mole, and in addition, is it subtracting? Sorry, this is. In addition, this is, the temperature, they're giving it to you in Celsius, right? Here, 20 degrees Celsius, but the temperature needs to be in Kelvin, so am I going to put 20 here? No, I'm going to put 293 because it's 273. Kelvin is 273 plus the Celsius, which in this case is 20. And then your delta S, they tell you, is 178. This is the change in entropy. So we got, they want you to calculate delta G, which is 46,000 minus what's 293 multiplied by 178? 52,000. 52,154. So subtract that by 46,000, or subtract that from 46,000, and your delta G is negative 6,154. Right? At temperature, calculate the change in Gibbs free energy. I don't think they're asking you to. I mean, you can do it as a whole number, 6,001. So you're going to be negative, right? 6,154. Is this going to, oh, sorry, put the negative right there. Is this going to be a spontaneous or non-spontaneous chemical reaction? Spontaneous. So this is going to be an exergonic, so you're releasing energy into the atmosphere because the final products have less energy than the initial products. So this is an exergonic chemical reaction. All right, take a look at this interesting little... This is actually easier than it thinks. It wants you to know how much carbon is being released into the atmosphere based on the metabolic activity of just the herbivores. First of all it is take the net in from the net, subtract what goes out from what comes in, and that'll give you what's left over. So the herbivores took in 100 grams of carbon from the grass, right? Because that's what they ate, they're herbivores. And what happened was some of the herbivores got eaten by predators, and so of the 100 grams of carbon in the sugar that was made by the grass, 8 grams went into the predators, right? So we start off with 100 that goes into the herbivores, right? So we have to subtract 8. And then some of the herbivores didn't get eaten by predators. Some of the herbivores just died, and then bacteria and fungus broke down their bodies and returned it back into the earth to be used by producers again. Subtracted by 50, and the remaining carbon that was used to make up their bodies was released as breathing carbon dioxide gas out through respiration. So that's going to be how much? You got 100 minus 58. You're going to have 42 left. So 42 grams per square meter. So if you think about it, I am made up of all the carbon from the food I eat. Okay? If you think of all of like human beings, some of us get eaten by dinosaurs, and the rest of us, we die and we get put back into the ground. The difference of all the carbon that was either eaten by a dinosaur or put into the ground is how much carbon we breathed out through cellular respiration into the atmosphere. What?
You have some.
Okay, we're going to combine rye grain, which has a 2N of 14, and wheat grain, which has a diploid chromosome number of 42. So diploid is 2N equals 42. We want to combine these two plants. Well, what we're going to do is we're going to take the pollen of one, right? We're going to take the gamete of one, and we're going to add it to the gamete of the other, and that's going to give us the diploid of the new organism, which is this triticale. So the gamete of this guy is going to have how many chromosomes? This is the rye, and this is the wheat. The gamete of this guy is going to have the gamete of something that is 12N, 2N equals 14. 7. So the pollen will have 7. But this guy's egg is going to have what? 21. So that means that the fertilized diploid of the new one is going to have 28, which equals 2N. Do I put 28 here? No, because it's asking me for the pollen grain of the new one. So the new one's diploid number is 28. When this guy goes through meiosis to make pollen, it's going to have 14. Round to the nearest whole number, 14. Got it?